I was asked to tell you or talk to you about what is basic science. And my approach is going to be a bit more, hopefully, discussion than lecture. And I have, about 20 slides, but I'll go through some of them fairly quickly. They're just to illustrate a couple of points. But my goal is to kind of pull back that curtain that a lot of people don't recognize what basic science is. This is a picture of Gregor Mendel. He was a basic scientist, and he, as you probably very well know, studied sweet peas. That's what the pictures of. And that has led to what we know as modern genetics. And so over the course of the next 20 minutes or so, I'm kind of hoping that I'll be able to answer a few of these questions that we brainstormed during a couple of conversations about how basic science informs clinical research and how clinical research informs basic science, how advocates inform basic science, and then a couple of myths and that type of thing related to the kind of research that the various people are doing that you've all mentioned. The first little bit is on some of the myths. And I have to say, I started this way when I first was in college and was looking to get a letter of recommendation to go to medical school, which I eventually never did. But I used to think the only kind of research that mattered was that which is directly impacting a patient. And one of the myths that I want to dispel is how we have a pipeline of things, and that will play a role. And one of the early myths is, how could study of plants impact what we study in cancer? And here, in this case, the rules that we commonly use in genetics were all studied in the sweet pea. That's one area in which basic science has informed what we do routinely nowadays in the clinic. Basic science is also known as fundamental research. It really, in biology, it's to understand how cells work. In physics, it's what is the fundamental particle, bosons, prions, and all of those types of things. And it's all based on understanding the fundamental problems so that we can study causation of things as opposed to just looking at correlation. And by its very nature, a word that you may see as you're talking with your paired up folks it's known as a reductionist kind of research. And what do I mean by reductionist? That is breaking things down so that we can control every variable, if possible, except for the one thing we want to test, so that we keep things as pristine as possible. And if you can do that, that means you can understand mechanism of how things are taking place. So you make an observation of some kind, you generate some kind of theory or hypothesis by asking that question. And based upon that hypothesis, you make a prediction, and then the experiments go into testing that prediction. If it's right, it supports your hypothesis, you go forward. If the prediction is wrong, that means you have to revise the hypothesis. So this is an iterative process. You keep going forward, 2 steps forward, 1 step back, and it takes time. I've been asked on a few occasions who are basic scientists, and the bullet just before the bottom are just a few of the different disciplines that are represented within this year's cohort of researchers, basic scientists, biochemists, microbiologists, immunologists, and so on. All of these folks are developing new tools, models, and techniques that'll be used. And a couple of things that I thought of, especially as I was preparing this is how we're hearing a lot, trust the scientists right now, especially with all the COVID related stuff. And yeah, yes, of course, I think all scientists have to be trusted, but you as consumers of science also need to realize that not all scientists agree. And that's really important because the data is not something you can disagree with. If it's the experiment's done properly, it's all correct. But the interpretation can be open to different things. And that's one of the things that you'll find when you're working with the basic scientists is we're kind of self-trained and we're selected for our ability to sort of ask questions that are skeptical. And so it's perfectly okay if you have skepticism about things that you're working with somebody else on. And I want to encourage you to feel empowered by that. So this is what my sort of definition of basic scientists or basic science is. And then what I think most people are familiar with is applied science, also kind of known as translational research or clinical research. And that's where the results found by basic scientists are applied to solve some kind of practical problem. And this is frequently the realm of engineers or physicians and that type of thing. So all of these are put together within the rubric. This slide is really simple, but it was the best one I could find, where basic research or preclinical research or foundational research is what's known as T0. And I have no idea what the T means. I'm going to say translation, but I don't know that for sure. And that then leads to the first in human type trials. If you're all pretty familiar, I believe, with phase 1 clinical trials. And that would be T1, then phase 2, phase 3, clinical trials where they just get bigger and bigger sets of patients is T2. Then rolling it out into the actual public goes T3 through T5. And T4 and T5 begin to be a bit more in the political realm because that deals with cost of medication, access, and the opportunity of everybody to get it. So, all of these are critical. They're all absolutely important, but each one plays a role. And but basic science and foundational research is really the first step. So this is what it's supposed to look like. But in actuality, it's kind of like mountains with valleys in between them. And those of you who've heard my colleague Scott Weir talk, there are 2 known valleys of death for things that are discovered in a laboratory and whether they're successful in getting into clinical practice. I think the last number I heard was 1 out of every 10,000 things that get to a clinical trial actually make it into routine medical care. And that's due to unexpected findings with regard to toxicity or side effects, or it just doesn't work when you have variables that you didn't realize were involved. One of the classic examples, it has nothing to do with cancer for unexpected side effects, is the little blue pill. It was designed originally to treat blood pressure. The men were experiencing an effect that was not expected, and their wives were also experiencing that effect. But it turned out in that case, the basic research could have predicted it if people had thought about why it was working the way it was. So even though the valley of death for clinical development of what is now known as Viagra was an unexpected side effect, that information made it repurposed for another disease. And I think that's kind of important. I pulled up some numbers and I just have to read them because I think it's worth knowing. The annual budget for the NIH right now is about $30 billion per year. And when you think about all the stuff we're talking about doing with recently the virus and the vaccines and things, this is a real drop in the bucket. And NIH is who funds almost all basic research in this country. I talked about these valleys of death, but I was thinking about it as you're trying to cross the desert and look for an oasis, but often you can see a mirage, and that's the bit of data that you look at, and it ends up being a mirage, and then you have to step back, revise the hypothesis, and move forward. So I like to think of things in analogies, and this is the best analogy I could come up with. Basic science on the left is like the fire hydrant that's hooked up to the hose, and the firefighter on the right is the doctor who's trying to treat the patient in this case. And if you don't have water going into the hose in the beginning, you don't have anything coming out at the end. And that's because there are leaks that spring up in this valley of death. Ultimately, if you have the leaks or the hose is not aimed in the right place, you end up with an uncontrollable fire. When I started my career, it was in the pharmaceutical industry. I know the evil pharmaceutical industry. There was a lot of stuff that went on, and I worked on a few drugs that actually are in the clinic, and I'll bet many of you have actually taken Motrin or that type of thing. I worked on that development team at one point. But there were things that we encountered, and I thought about a few of them. Toxicity, where side effects, the expense, it would cost too much both for the patient to take it, but also too much to manufacture it for a particular price, for a price point. The lack of patent protection. It costs about $2 to $3 billion to develop any drug that's approved by the FDA, and the company's got to be able to make some of that up, and they can only do that during the exclusive time for a patent. Stability. A lot of drugs are great if you can handle them in pristine ways, a lot like the Pfizer vaccine right now. You have to keep it at ultra low temperatures. And because COVID has been so devastating, people are willing to put up with that. But if we weren't experiencing 500,000 deaths, probably the Pfizer vaccine and even Moderna probably would not have been approved. And probably the single dose things that can be held at room temperature would have been. So that's my political statement, but I think hopefully you're seeing a pattern with this stuff. Another little bit about basic science. So I'm using KU here. And in the middle, a bunch of drug companies, and these are the key players in oncology. And then on the right are the clinicians. And nowadays, almost all basic research is being done in universities. Although each of these companies has some kind of basic research going on in it, typically the model nowadays is that grants fund research at universities, medical schools, and so on. And then when they get to be a point where people are publishing something and it becomes recognized, then the pharmaceutical companies or biotechs buy that technology, bring it in, and then pay a lot of research, somewhere close to a couple $100 billion a year in development costs. And then they develop it to the point of handing it off to the clinicians. So this is sort of the pipeline. Again, it's a continuation, a continuum. And the basic scientists, I include myself in this, are really at the very beginning of the process, and very, very, very few of us are able to really participate in something that goes all the way to the clinic. But almost all of us contribute something that makes a discovery at one place become applicable. And I'll give a couple of examples of that. I thought I'd spend a couple of minutes talking about some myths and reality. And a lot of people, especially when you watch television nowadays, look at labs like this one in the upper right. Oops, don't look at this yet. In the upper right, it's like what you see on NCIS, where they've got 10,000 square feet, one person who's got 100 pieces of equipment, and they operate every one of them to perfection. That's not the case. It's typically a little bit more messy, like this one in the upper left. And this one's actually not as dirty as some labs I've seen. Typically, it's a little bit more crowded, a lot like this office with graduate students, probably graduate students or postdocs. In this case, we don't have private offices or anything like that. So I just think it's important that we let people know what it's really like. And I can tell you that Sarah asked that a few of us in my lab, we're going to be making a couple of videos to show some things from 2 or 3 labs. And I'm partly throwing in that message as an Easter egg for Sarah. We were talking about it today, but looking forward to giving tours when we can open back up and gather together, because I think it's kind of fun to see some of this stuff. A couple of other myths. I just love it when they'll get a sample of from a crime scene on those shows, and they send the sample in, and then like a few hours later, they have a whole genome sequencing. That typically takes 2 to 3 days, even in the best labs. And Children's Mercy happens to have one of those. But almost always, it usually takes a week or 2 to get that kind of data. And, you know, just those kind of myths. Here I'm showing our friends at the Big Bang Theory. And so the first thing is to recognize all basic scientists are a little bit geeky. I'm amongst that group. I often joke that we're all somewhere on the autism spectrum, where we have quirky senses of humor, and we speak in details that seem really weird and arcane to the non-geek amongst us. And it's not that basic scientists don't want to communicate, but it's often easier to communicate in our own native language. And that's one of the places where you'll come in, is to try and help us talk in English in this country. But it's really important. And when we're doing basic research, I was trying to think of an analogy that I think all of us could relate to, and you'll have to tell me whether this works for you. But we're trying to put together a jigsaw puzzle, but we don't have the box that it came in. So we're not even sure what the picture is supposed to look like, yet we're trying to fit all these pieces together so that we can come up with a final picture. So I think that's important. Another thing that I will tell you, even though almost all my friends and I like to watch Big Bang Theory, one of the things that I heard probably 20 or 30 times was how unrealistic the show was with regard to grants. It was hilarious that they would say, oh, I'm going to go make a presentation in 10 minutes. to the dean, and the dean never controls that kind of money. And I'll be able to get a grant. In fact, grants are a pain in the rear end. That was not the best word I could have chosen, but I was tried to make this PG. Each grant that I've written takes about 2 months, almost full time, to write. We submit it. It undergoes a review by experts. And the time from our submitting it to when they review it is typically about 3 months. It then goes to a second tier. And this is like in a company where you have people at the mid-manager level, but then the next level up, they're looking at the whole portfolio. You don't want to put all your eggs in one basket. And this council actually looks at the overall portfolio to make sure that we're not ignoring viruses to the exclusion of cancer, or doing only cancer research when heart disease is important. And then once council, if they approve it, it takes another 3 months before a grant is activated. So we're talking from start to finish, in the best case scenario, of 11 months to a year. And currently, the way we feel is that we're banging our head against a wall, because the pay line at the National Cancer Institute is about 6%. That's 6% of grants that are deemed worthy enough to get funding if there was unlimited money. Only 6% get paid. The American Cancer Society, it's about 5%. And I don't know what it is for some of the other places, but they're typically much lower. A couple of things just worth giving you information about. For every dollar that we bring in at KU, I'll just use us as an example. So if I get a $100 grant, the agency typically is providing $54 in overhead costs. At some institutions, they actually get more in overhead than the grant itself. So this becomes an expensive venture pretty quickly. And I put this down here for compliance. This is one of my personal pet peeves. And apologies for bringing this in, but it drives me crazy. Every year I spend a full week dealing with compliance-related issues, taking a bunch of tests and things like that, where they're trying more to trick us than to ask us things that are really important. I'll get to that if you care. But this kind of funding that I was talking about, I'm using that same continuum of T0 through, this goes through T1 and T2, but we're T4, 3, 4, and 5, and so on. You get different kinds of agencies funding this at different levels. You need venture capital, grant funding for the early steps that we deal with. But later on, it tends to be stock offerings and that type of thing. So what are these grants? Well, I just want to show you a couple of quick things here, just to give you an idea of costs of different things. And I can say in my lab, I have every piece of this equipment except this mass spectrometer in my lab. I don't have a cell sorter, so I don't have these two, but this is the kind of typical lab setup that we have. And that's not including the people who have to run it, that type of thing. I put this one in on the far right. These are sterile pipettes. They cost $1 each. A typical experiment would take 30, 40. And so you start doing that every day for each person, and you can see how things just start to add up. I didn't put mice in here, but for those of you who are curious about it, the range of price for mice is somewhere between $25 and $1,000 each. And then when we get them, they cost $1 a day for room and board to keep the mice healthy and happy. And here are millions of dollars cost to recruit somebody at these various levels. So just to let you know roughly what these kind of things cost. And this is to set people up for 2 or 3, 4 years to do things. All right, so why should you care? I'm going to do 6 vignettes here. And this will be really quick, but they're some of the six of the most commonly recognized Nobel Prizes. In the year 2000, I believe it was, these 3 folks got the Nobel Prize for studying how cancer cells divide. I mentioned earlier that plants could have an influence. They did all of their work in yeast, common yeast that you use to have bread rise. And it's important to recognize that basic research sometimes uses simple models so that we can get through it. A few years later, in 2009, these 3 studied how cancer cells die. And that's important because it's one of the mechanisms that you get cancer. They don't always divide more rapidly, they just don't die enough. And that became recognized in the early 2000s as something that we should target. And drug resistance is one of the ways in which that's helping understand why people have therapies that fail for them. How cells secrete things. I'm going to put a little PR. This fellow, Randy Sheckman, is actually giving a seminar here at KU in I believe about a month. If you want, we can get you a link if you wanted to hear it. It's going to be a very geeky science talk, but we're going to have a Nobel laureate here. Anyway, these 3 studied how cancer cells secrete things, and this is the basis by which investigators are now developing what's known as the liquid biopsy, to be able to identify markers in the blood or cells that have entered the blood, and how those can be used to inform the physician who's treating a patient. I'm very proud to say Jim Allison, who's here on the right, was one of my professors when I was in graduate school. And the work that he got the Nobel Prize for 2-3 years ago, he was starting to talk about that back when I was a graduate student. So it took him a long time, along with Tasuku Honjo. But this is the basis for all the immune therapies that are currently approved in the clinic. 2 years ago, Greg Semenza, Bill Kalin, Peter Ratcliffe were studying how cells sense the presence or absence of oxygen. And you would think, wow, we should know that already. Well, interestingly enough, this turns out to be that these sensing mechanisms are actually responsible for controlling the development of kidney cancer. Very unexpected way in which those 2 things connected, but it was pure basic science in this case where they found it. And then last year, 2 prominent women, Emmanuelle Charpentier and Jennifer Doudna, developed a technique called CRISPR. And this is the basis for gene therapies that are being developed in the clinic. And this is all very basic science. So hopefully that just gives you a sense of how exciting it can be, even with the geek factor thrown in. And seeing those possibilities is what makes basic science exciting. So I want to sort of conclude with what roles you can play in basic science. First of all, at the top right, I found this just sort of nosing around the internet. And the role of a research advocate is to change what is into what it should be. I just thought that was the best possible definition. And that involves teaching and learning and working together with each other, and communicating effectively. this is on the left, bottom left. Cheryl has, I heard this quote, but she helped me find this picture. And, you know, geeky scientists, we talk in our own jargon, and we have to be able to explain things to other people. And that's where you come in and give us a reality check so that we can accelerate the progress of what we're doing. And I will say I worked in cancer research for over 20 years before I really heard the word lymphedema as an example of something that women with breast cancer mastectomies had undergone. And it was because a few basic scientists eventually heard that they started working on ways to treat it. And in the last 5-7 years, lymph node transplants have taken place, and it's being commonly done in Europe. And that is really helpful for patients to minimize that kind of stuff. And they did all of that in mouse. So learning and teaching and brainstorming and giving back and forth. So ultimately, this is the tapestry that we put together, working together, pushing each other to make progress. So I tried to do it in about 20 minutes. I did 22, so it wasn't too bad. I'm going to stop sharing so I can see you and try, if there were questions, to help make some sense of it. So, oh my gosh, lots of chat. Okay. Okay.
Yeah, I think, and Cheryl, I think you had some questions that people echoed. So do you want to ask Danny your question specifically?
I typed a lot of them privately to Danny too.
Okay.
But, so which one? Okay, one of them I said, Danny, was the most important one at this point. You mentioned the cost to recruit. Is that salary?
Not usually. That is an addition to salary.
So what goes into the cost to recruit?
Giving somebody enough money to hire a technician or a student for 2 or 3 years so that they can make progress, collect data to apply for grants, buying equipment, the supplies, a token amount to go to a meeting or two a year to interact with other scientists, that type of thing. So, and the more senior you get, the more you get because you've got a track record to prove it. But it's still $1.5 million for somebody straight out of their graduate and postdoctoral training.
So you didn't get $1.5 million salary, right?
Oh gosh, no. Broderick gets that much, but I don't.
And it all goes to Sarah.
So, no, salaries for academics mostly range in the $80,000 to $200,000 a year range. It varies quite a bit. There are some people paid a lot more than that, but that's a range from starting to somebody who's been doing it for about 20 years.
Someone who's got the grants that are being pulled in.
Yeah. So one of the things I think I should have said, and by way of the way you asked that question, most people think that most of my salary comes from KU. It comes from grants. I am basically an entrepreneur operating a small business. And if I don't bring in grants, my salary is cut by whatever I'm not bringing in. So when I was talking about grants, that is that's the key thing for people to know.
And I don't know, I can't remember if you specifically referred to this, Danny, but out of At one point I heard that it was like 1 out of 10 grants gets funded. Is that still kind of what we're looking at?
It's less than 1 out of 10 now. It's closer to 1 out of 20.
So it's not a very big chance of being funded. And so you have to submit a lot of grants.
Yes, you've got to, yeah, on February 5th, I submitted 5.
And we're crossing our fingers, all of them get chosen, right?
From your mouth to God's ears.
And the only other one that I was asking about that I think someone echoed, Tracy did, compliance. You may need to explain a bit more about this. Compliance for what? And who in the heck is judging whether you're compliant or not, and for what purpose?
Okay, yeah, and I apologize for not being clear with that. It was part of my jargon. But every year I have to take a series of tests that prove my competence to work with mice. And it's like being renewed for a clinician. They have to redo boards every few years. My quibble with most of that compliance stuff is not that they're asking those questions. I want to be competent, but I'll give you a better one. Every year I have to take tests to use radioactive materials in the lab, because that's how we have to do some experiments. And they start asking questions about building dirty bombs. It's irrelevant. I'm not going to build a dirty bomb, but they ask those kind of questions. And then they will ask a question that basically is memorizing some trivial point that I could look up in 5 minutes, but I don't use a particular isotope, so it's not relevant to me to memorize how long it lasts. So that's my quibble with it. It's not the process, but it takes about 60 hours a year, every year. So that's 1 week a year to do it.
But every kind of scientist, whether it's basic or clinical, has to go through different kinds of compliance tests that
Yes. Yeah.
Can be very taxing, and that's a big issue.
So if you don't mind, I'm going to go through the chats, and since I can see them, Sarah, I'll just read through them. Okay, Vicky, you said I forgot to add you're not paired with someone. I am pretty new. That's okay. You will be. We're looking forward to that. Charlene, non-Hodgkin's, 5-year with Brandon Dukoski. He's an immunologist. Sorry the tech issues were there. Cheryl's with Sufi Thomas. She's a head and neck cancer person. Broderick, can we trust all scientists? No. But Henry Ford said something like faith, you can have faith, but you need to bring data. And you have every right to question the data. And if it doesn't fit with what they're saying, don't trust them.
Well, and Danny, thank you for your presentation. And of course, I go a little deeper than that. So we know that there are many physicians that were lauded as pioneers. For example, J. Marion Sims, pioneer for gynecology, but yet his practices were on enslaved women and not using anesthesia. And yet many of the things that he studied and perfected is currently being used. So my concern is, one, can we trust them? And then two, as you talked about recruitment, what level of intentionality or is there with recruiting scientists of color?
Okay, so there are about 8 questions built in there.
That's what happens when we go on the road together. I get to know you, sir.
A lot of things have been built in since the unethical behavior of people doing such things came to light. So, I mean, besides Sims, we'll look at the guys who did all the stuff to the poor Jews during the Holocaust. That's another example. The Tuskegee experiments, another example. As things happen, the majority identify those and say, we need to fix it. And I'm looking at John here. If I recall, you're a dentist, correct, John? Okay. John's probably seen things take place in his dental practice and teaching that were considered shady. I won't call them unethical because I don't know of any unethical dentistry. There probably is, but I don't know it. And as that happened, people put things into place. I use mice in my lab's research. There are people who believe we should never use a mouse. And I would say, I know of nobody who does work with mice who wouldn't give it up in an instant if we had things that worked as well. But because of looking at the ethics, now there are a whole bunch of things to be compliant with. We need to give them anesthesias and things like that so that we're not making them hurt, and testing through those things. What was the other new thing, the other thing you had at the end?
Recruitment of people of color.
People of color. That, in fact, I hate to admit this to you, I'm now sort of somewhat responsible for that within the Cancer Center. In terms of education, my role is to make sure that we have diversity both in our students as well as the faculty, and that underrepresented people in science, regardless, are getting the best possible mentoring at all levels. If we have a little bit of time, Broderick, so that I can get through these others, would you remind me at the end, and I'll tell you a story of how I got involved in doing some of this when I was at Upjohn, but I think some of these other questions are more direct.
Actually, Danny, that sounds like a Zoom call coming up later this week. How about that?
Okay. So, okay. Becky said she worked in pharma for a while and looks familiar. So at least it didn't look foreign. Yay. Charlene, thank you for that. D had to log off. Compliance, echo Cheryl. Cost to recruit is the salary.
I think we covered most of the questions. Danny, one thing I did want you to discuss too is, can you tell us kind of how many labs or, I mean, how many in the cancer biology program, kind of what are we looking at? I mean, staff wise, and, you know, who we may have the opportunity to work with.
Right now, I don't know the exact number, but there are about 70 labs in the cancer biology program. That's the basic research. And each lab has between 3 and 10 people in it. So we're talking several hundred people who are involved.
Great, that's helpful.
So,
does anybody else have any other questions before we wrap things up?
I was wondering, did much of that slow down during COVID, especially given the close quarters that you're normally in?
Oh, it slowed things down a lot. My lab, we probably lost 18 months of momentum because in order to deal with social distancing and that type of thing, for reasons that we all understood, we had to actually euthanize almost all of our mice, and they're genetically engineered. So once we had the sanctions lifted, and I'm using the word sanctions, but the recommendation, then we had to start breeding the mice to bring the colony up to size so that we could do an experiment. And we actually lost 2 of our strains and had to recover them from liquid nitrogen, much like fertilized eggs. We had frozen fertilized mouse eggs back and actually had to implant those. And we just got a couple of those back up. So it's going to take another year of getting those. So it's a great question. And I was listening to you, but I couldn't figure out who it was. And then somebody else's thing popped up to say who was asking the question. So
that was Kathy's question. And then Katie just wanted you to touch on the value of your young investigators.
Their value is immeasurable. First, let me just say, in the university setting, we, and I'm saying this in a big way, a little bit over the top, we don't have a career path for people to stay in research. It's by and large, we're training people who then go on to do things. If you don't get your PhD, you're really stuck and don't get to move up in the salary scale to really do things well. Pharma does a much better job of that. Where do I find young people? They're not jaded by having heard things. And so they're more willing to ask the trivial question that sometimes it's like, why didn't I think of that? There's an energy that comes with youth that is lost on people who've been doing this for a long time. When you have people who are getting together and they're talking with each other and they're excited about it, it creates a buzz and an energy. And there's a lot more social interaction between young people. And so frequently they're talking to folks in other disciplines and saying, oh, I talked to my friend and they're doing this very cool thing. Did you ever think about bringing that into our lab? And, no, I've never thought about it, but you got a good idea there. So I've been very fortunate, and one thing I think, again, I think if I recall, I met John at the last Alan Alda thing, so we never interacted before that. But I believe you were on the faculty, UMKC.
Yeah.
So I think you can probably share this a little bit, but there's just that bit of energy that comes from them, and then they'll come in and they'll start questioning, and about the time they're ready to graduate, they hate me as a mentor. Hate is probably the nicest word I could use. But about a year or 2 after they leave, most of them say, thank you. And that's heartwarming in a big way. And I will say, Cheryl has been teaching in my cancer biology class, the intro class. And one of the things, and maybe more of you can help do it in some next year, but one of the things we have them do is write their research abstract in lay language. And when Cheryl and I first started talking about this, I said, grade them hard. It's okay to get an A- in school, but when you write a bad lay abstract and you're applying for a $2 million grant, you don't want people saying, I don't get it. It's got to be really clear. And she does that beautifully. So I'm not just saying that because she's on this call. I've said it other times. But they don't like her immediately after she's graded those. But they love her when they've applied for a fellowship and they get a really good comment. So they really love her all the time, but they don't like those comments. And Cheryl, I can see you smiling, but you have to know it's true.
Well, and that's good advice for those of you who are paired with our basic scientists. You need to be kind of tough on them, and their lay abstracts are not going to be great at first, and so that's why we're so thankful that you guys are here to be that person for them. So, well, it looks like we're kind of wrapping up here, but if you guys have any additional questions for Danny, please just email me and I can forward them to him and we can get those answered. But Danny, thank you very much for your time and helping us understand this a little bit better.
Thank you.
Yay.
All
Thanks,
right.
Danny.
Thanks for the very kind comments. I don't know.
Yes, yeah, definitely read those. Those are great. All
And
right.
I also share that if you ever have an opportunity to go on the road with Danny, he's a good guy to travel with.
Likewise, my friend.
Yeah, let's travel again sometime soon.
Yeah, we're all hoping we can do that.
Now let's travel.
Yes. Yeah, what is that?
From the living room to the dining room?
Yeah.
Oh,
Well, I have to admit it's more comfortable from the living room to the dining room.
Yeah.
than an airplane.
not as much fun, but very much more comfortable.
So,
All right, everybody. Have a good Monday. We'll see you later.
You.
Thanks,
Thanks,
Cheryl.
everyone.
Thanks, Danny.
see you Friday.
Bye.